So in this whole trip from the upper respiratory portion to the lower respiratory portion is in fact the whole logic to reach the lung and in order to exchange the gas. So this whole respiratory epithelium, we can see, has done two things. It has entrapped all the through the mucus all the pathogens, all the foreign particles. It has moistened the airway. It has moistened and warmed up the airway through the function of the upper respiratory tract, in order through the function of the capillaries, of course, through the function of the heat, let's say, heating and moistening the whole air. So all of this function, all of these logic was done in order to reach the lung and to have this exchange. Now, I told you before there are two different classifications, the anatomical ones and the histological ones. The anatomical ones do not necessarily let's say reflect the function, but this one and the histological one, the conducting respiratory portion, the classification does. So we'll see how the bronchi break up to bronchioles, to terminal bronchioles, respiratory bronchioles, alveolar ducts and sacs, and lastly the alveoli. Of course, the cells that do all the work, that do all this exchange of gases, is found within the alveoli. We'll see which cells are that. But before we get there, we should really understand and comprehend the way that we go from the respiratory epithelium to the very, very small, simple squamous epithelium of the alveoli. As I told you before, and again many times, the whole ductular system, because this is practically a duct, this is a very, very large duct in a very, let's say, general idea. We're gonna have this whole starting from a big type of epithelium for the columnar, let's say in this case, to reaching the very, the tiny one throughout the whole leveling and gradual, let's say, smalling in size cells. So in this here, shrinking in size cells. So we start from the bronchi. We can find the typical, the respiratory epithelium, along with the prominent bands of smooth muscle we saw, for example, in the trachea, and of course the irregular hyaline cartilage plates after, of course, the trachea, and after we're right here in the bronchi, the primary bronchi, the right and left. So in this case, we're going to find again the same, the features that I just talked about, along with irregular hyaline cartilage plates, not fully concentric as we can see already in the trachea. So the point, of course, of this function is again to conduct the air with the same logic as we discussed before of the conducting portion. As we break into smaller parts, the bronchioles, how do we differentiate the bronchioles from the bronchi? The first element, the first clue, easy to see, is the absence of cartilage, the first one, because we can see this is a very useful table from Junqueira. You should absolutely use it in order to study and to really comprehend the differences. As we can see here, it actually points out that there is no cartilage and there is a circular layer of smooth muscle. So this is the first difference. The second one is the change of the epithelium. We had from pseudostratified, and we proceed on to the short columnar to cuboidal cells. Plus, we find one very unique name here, the Clara cell, or also called the club cell. Now, the Clara cells are cells that are, again, typically speaking, columnar when it comes to size. Of course, there are no cilia in this cell, and the functions of the cell are multiple. For the first one is to produce surfactant. We'll go through the logic of the surfactant in just a bit later on. But remember this is a lipoprotein, let's say, component that is being produced constantly, and it's necessary, of course, within the whole point of the respiratory system. So production of surfactant, number one. The second one is that it produces antimicrobial peptides. This is, of course, an immune-related function. Furthermore, we have these cells also play a role in the mitosis. These are also considered to be stem cells for other cells we can find in the whole length and the whole mucosa of this specific portion. And lastly, of course, they have a detoxificatory function. They detoxify different sort of toxins that we can find along the line of the bronchioles, and this is done, of course, by the extensive smooth endoplasmic reticulum we can find within the Clara cells. We'll see a picture of them in just a bit. So after, of course, the next branching out of the bronchioles is the terminal bronchiole. Here, the logic, of course, is again to shrink the size of the cell and, of course, let's say smoothen and let's say thin and reduce the amount of smooth muscles as we go. Of course, as what's the logic? The logic, of course, says that after columnar, low columnar to cuboidal, we're gonna find simply only cuboidal epithelia. Of course, still ciliated in this case, with clara cells present in the whole mucosa. And of course, we have the change to the thin and incomplete layer of the smooth muscle. So this is the last exact point of the conducting portion. We finish with the conducting portion in the terminal bronchioles. And of course, it's very, very easy to remember exactly where we finish. And this, of course, why I put the red line here, exactly here, to denote the ending and the beginning of the two different portions. And the first and easiest way to remember, of course, the ending point is that exactly after terminal bronchioles, we have the respiratory bronchioles. And of course, as the name suggests, and I told you already, the function of respiration is being done by these alveoli, the points of the cells within the alveoli. These cells are the pneumocytes. Type 1 and type 2 pneumocytes are present in the alveoli. And of course, the cells that do have a role in this that do have the function of exchange, of gas exchange, is a type 1 pneumocyte. We'll discuss this further on in a bit. But the logic is here we're going to start to see exactly that, exactly the alveoli, scattered alveoli. We're going to see scattered, let's say, simple squamous epithelia along the whole line of the mucosa, in this case of the respiratory bronchioles. So we're going to find here a mixture of cuboidal, short cuboidal cells, still ciliated, still clara cells, with very, very few and scattered simple squamous epithelia, or also called in this case specifically type 1 pneumocytes present in the alveoli. So this is exactly where we can easily understand the differences of where one stops or when the one ends. And here we're going to start seeing way, way fewer, almost to no, very, very sparse and very, very few amount of smooth muscle cells. And here, this is the first point where we start having gas exchange. Then moving on from the respiratory bronchioles to the alveolar ducts and sacs, here we start to see exactly this reducing in the balance and this shift in the balance from a lot of cuboidal to very, very few alveoli, to simple squamous pneumocytes, to a lot of alveoli, a lot of presence of simple squamous epithelia, and the very, very low amount of cuboidal cells present in these ducts and sacs. And lastly, we go to the star of the show, the whole point, the whole rationale, the whole structure that we saw, from the epiglottis to the trachea to the bands of cartilage to the bronchi, bronchioles, terminal bronchioles, respiratory, and the alveolar duct, is the alveolus. Here the alveoli, the alveolus 1, alveoli many, this is exactly the point of the gas exchange. Here we have no presence of muscle, no presence of cartilage. As of course you can see, the cartilage stops. The cartilage stops exactly in the point of the bronchi and never to be seen again. And of course here we're going to find this alveolar part. We're going to find no presence of smooth muscle. We're going to simply find only elastic articular fibers. These are very important. You should remember them because there are many pathologies inclined and many pathologies implicated with this exact balance of elastic and articular fibers when it comes to the support of the simple squamous epithelia. And of course this is where we can find the location of the gas exchange. So here we're going to find type 1 pneumocyte, which is the cell responsible for this exchange. We're going to find type 2 pneumocyte. These are the cells that produce surfactants, and these are very important cells. And lastly, we're going to see the dust cells. The dust cells is nothing more than a fancy name for the alveolar macrophages. So the whole gas exchange procedure should be discussed in the end in order to really understand first the histological section, the histological technique, the histological rationale and morphology, and then we're going to move on latest to the function to really go through this unique and wonderful gas exchange and the whole structure of this logic. So let's start seeing these things in the macroscope. So this is the section we should actually discuss. The first point that you can actually see is, even from the distance, is this very, very big band of cartilage. And I told you before, because as long as we have cartilage dispersed with a very, very characteristic spongy appearance of the lung, this is a very, very nice way to really lock in and understand that this is exactly the bronchi, the bronchi. This is a bronchus. So if we zoom in, we're going to find exactly the features that we discussed about. first we're gonna find the epithelium, the respiratory epithelium exactly right here, the typical ciliated structure along with the lines of the mucous membranes. Exactly beneath we're gonna find the typical structure of the basal membrane, the very thick basal membrane, as you can easily see. Exactly in the base, the basal membrane is very thick, exactly beneath the basal cells. Then again, typically speaking, the lamina propria. Then we start with a very short amount, but the continual, let's say, band of smooth muscle, and beneath we start to see exactly the typical features of the hyaline cartilage, and this is a very prominent one exactly right here. So practically whenever you see the cartilage in this level of section, in this length of this height, let's say, of the section, you should actually easily identify that this is and understand that this is the primary bronchus. So exactly after this, we're gonna see the bronchiole. The bronchiole, as you remember, we go from the pseudostratified to the columnar, to low columnar, to cuboidal, and let's actually see them specifically. As we can see, there is no presence of cartilage or surrounding it. Easily to understand, this is a whole band of mucus present in the whole lumen of the mucosa. So you can see exactly here the very nice columnar cells, shorter columnar cells present exactly right here, and the structure is exactly the same, basal membrane present. Again, there is a band of scattered there's not bands, like scattered columnar, smooth muscle cells present beneath the lamina propria, and that's pretty much it, the structure of the primary bronchiole. Now it's important to notice and to find specifically the Clara cells. Now it's not very easy to find the Clara cells present in the section. Of course, the easiest way is to start looking for sections that do not have the cilia present. Unfortunately, this section does not help us because we have the mucus present in the center and there are many artifacts present in the whole section. But we can see one in the presentation right here. This is taken from another book. We can easily see the presence of the cilia on the top, and the Clara cells have again similar morphology but no presence of cilia anywhere in their apical domain and apical part. So then we're going to find the terminal bronchioles. The terminal bronchioles, again, the difference is we went from the respiratory epithelium to the simple columnar to cuboidal, and now we start seeing exactly short cuboidal, typical more cuboidal presence of the cells. But of course, still we're going to find presence of cilia. For example, this is a very nice section where we can find cilia intermixed with mucous membrane, let's be honest. And we can find the scattered loose, smooth muscles present, and of course surrounding it are the very small amount of connective tissue surrounding and covering the whole section. Now this is the last point again of the conducting portion. Exactly after that, we're going to start seeing the respiratory bronchioles. So how do we identify the respiratory bronchioles? Well, as you can see easily in the picture right here, we can have cuboidal cells. I already mentioned that we're going from cuboidal to squamous. So again, the terminal bronchiole had cuboidal cells, ciliated cuboidal cells, and now we'll start with the respiratory portion, which is exactly the addition to this cuboidal part of some simple squamous epithelia. So you should start looking for areas that have both of mucosal lines, of course, that have both cuboidal and simple squamous. And as you can see right here, there is a very nice portion of exactly that. So in this section, you can actually see the very nice cuboidal cells present right here. And as you can see, the continuation of the mucosa, we start seeing a very few scattered and very small amount of simple squamous epithelia right here. These are the type 1 pneumocytes, and this is exactly where we start with the respiratory portion of the whole lung. And as we move on to as we start moving on, we should actually go from the terminal bronchioles, from the respiratory bronchioles to the alveolar sacs and ducts. So in order to easily identify the difference between the ducts and the sacs, well, I'll be honest with you, it's not that clear and not that very, let's say, distinct areas histologically speaking. One trick to do it is, of course, with the diameter and the size of the opening. For example, as you can start seeing, this is the typical alveoli. For example, this is the alveoli, the smallest branch. And now you should compare based on this size a bit bigger, more alveoli present is the sac. For example, let's start seeing a sac. This is an alveolar sac because we can find many alveoli present right here. These small areas are the alveoli. This is exactly a very nice feature, very nice example of the alveoli. Also here, this is a big one maybe. So this is pretty much the shape and the size. So if you start seeing, let's say, portions and branching outs of few branching outs within this area, this should be a sac. Now, if you see bigger areas, for example, this one right here, this should be the alveolar duct. But the logic, the point is exactly the same. The point is going to be that it's going to be covered with simple squamous epithelia, of course, lined out throughout the whole layer. And distinguishing from between the sacs and the ducts is only a matter of size, only a matter of opening of the lumen and diameter and opening of the, let's say, lucent area. So let's zoom in and start seeing specifically the alveoli. So this area, as you can see, is filled with simple squamous epithelia and small spherical nuclei, which correspond to either two different types of cells that we find also present in the alveoli: the type 2 pneumocyte that produces the surfactant, and the dust cell, which is nothing more than just the alveolar macrophage. So this is the whole structure of the lung. Now let's start talking about the function, of course, and the exchange between, and how does this exchange work. So as I told you before, type 1 pneumocyte is the one that does very big part of the job between the diffusion and exchange of CO2 to oxygen. So this barrier is called the blood-air barrier, and as we already mentioned, any blood something barrier always has the, of course, the endothelial cell, of course, that covers up the capillary, the common diffused basal membrane, and the other cell. So here we have the blood-air barrier. So of course we have the type 1 pneumocyte on the bottom. It's on the bottom, type 1 pneumocyte. Exactly above it, the basal membrane, the common fused basal membrane, and the other portion, that last part, is the typical endothelial cell of the capillary. So this is the real way, the real, let's say, macroscopical picture understanding of how this barrier works. So with this exchange, from using, of course, both features of the erythrocytes and the exchange of the lucent, let's say, the release of oxygen from the of carbon dioxide from the erythrocytes, and the diffusion of oxygen from the air, from the, of course, the lumen of the alveolus to the erythrocyte, we have the exchange of these two gases. This is the logic. Now let's talk a bit about the other cells. Important to understand, important to notice that we have these alveolar pores. Now, why is that? Because as you can see, of course, the alveolus is nothing more than just a fancy sac. The point is that if you do not have continuous and non-interrupted connections of all the alveoli as a unit, then you have different pressures from one side to the other, and you lose the functionality and the proper synchronized and organized fashion and manner of the alveoli. I know that this structure, the structure of the lung has many names, but it's really easy to understand it when you start from the bottom to the top. So if you start with the basic unit of gas exchange, which is the alveoli itself, we have the alveolus. A bit above it, we're going to find the alveolar sac. After the sac, we have the alveolar duct. After the alveolar duct, we have the respiratory bronchiole. After the respiratory bronchiole, we find the terminal bronchiole. After the terminal bronchiole, we're going to find the bronchiole itself, and after the bronchiole we're going to find the bronchi. Don't forget the bronchiole, any with all, it means small something. So bronchiole is the smaller bronchi. So exactly, at least when I was shooting, it was really helpful to me to remember the bottom up. Other students prefer it bottom down. It's simply a matter of taste and a matter of let's say preference. So one point which we mentioned is the alveolar pool and the alveolar pores and the septum. The interalveolar septum is, of course, the site, the area between the capillary and actually just the connective tissue surrounding the base, the blood-air barrier. For example, as you can see, this is the very nice and clear slide and clear picture of exactly this point. And on the left hand, we're going to find this alveolar connective tissue, which is also called the interalveolar septum. Now, why do we care about this? Well, because the interalveolar septum is again an area that we can find altered and changed in case of pathologies. You'll see exactly more of these pictures in pathanatomy. This is a bit beyond the scope of histology, but just an idea of why you should know exactly what it is and where we can find it. So I already talked and mentioned about the Clara cells that produce surfactant. I talked about the type U pneumocytes that also produce surfactant, but what is this surfactant? So this is the surfactant is a product that is being produced by the type 2 pneumocyte. By the most amount of this surfactant is produced by the type 2 pneumocyte. Now, the type 2 pneumocyte has dual function. Of course, one is to produce surfactant. The second is that it plays it's a stem cell in that way that it actually, whenever there's destruction of the type 1 pneumocyte, type 2 pneumocyte will differentiate into the type 1 and cover up the loss and support the whole structure. So this is exactly the rationale of how the type 2 works. It produces always a protein and produces the protein that is present in the surfactant, produces the lipids, of course, composes the lipids of the structure. Then it actually modifies the contents in the Golgi apparatus and produces this lamellar body, which is the content, of course, of the surfactant. As you can see, there is the fusion between the particles of the proteins and the small lamellar body, which makes the phospholipids. So the point of the surfactant is to reduce surface tension. Now, what is that and why would we care so much about that? Well, as we know from the law of Laplace, we have the tendency of small areas, of small spheres, to collapse when we have this altering pressure in and out. As we can understand that this is the exchange of the gas between the area of a very, very small sphere. So this is a very nice representation of the whole structure, the conducting airway in, let's say, in very practically graphed, and the alveolus is a sphere that contains, they have the exchange of gas. Now, when we have when we expectorate air, when we exhale air, after exhalation, we have this tendency of air to go outwards. When we have this loss of volume within the alveolus, now this alveolus tends to collapse because the content, of course, that held the diameter open tends to collapse, and this pressure can be calculated by the law of Laplace. And this is one you should actually know, because many teachers and many academics ask for this to at least they want you to understand the difference in the radius, the diameter, that the diameter is reversely proportional to the pressure and vice versa. So what's the point though? How does the surfactant actually change anything? Well, because we have, of course, this area is a moistened area. This is where water is present in small amounts, of course. Don't imagine major amounts, then you will suffocate. So of course we have the small amounts of water in between these areas. So in order to break the surface tension, in order to release and to reduce surface tension, we have the surfactant, which is the phospholipid content. It plays again the typical logic of this, let's say, this hydrophilic and hydrophobic parts, that in this way they contain the water and they break this whole continual, let's say, circle of water, and break the cohesion between the water molecules. So imagine that that the surfactants are barriers between the different water molecules and do not allow for the whole continuity of the water molecule, because of course the surface tension, in simple terms, can be explained as the tendency of the water molecules to stick together. So if you have this tendency of water to stick together after expectoration, and the whole lumen the whole periphery is encircled with water, then the surface tension will actually maintain and shrink even more the alveolus. As a consequence, we have this collapse of alveolus, which of course renders it non-functional. So this is the point of the function of the surfactant, to reduce surface tension and allow for the diameter to maintain itself more or less, always open of course, and to maintain the diameter more or less in a similar size and number. that's it. That's what I want to discuss. Thank you very much.